Welcome to the Molecular Therapy Presents Targets and Biomarkers webinar. Today's seminar is a panel discussion where we will discuss target discovery strategies in cancer immunotherapy, the emerging role of tissue-derived extracellular vesicles for disease target identification, and the utilization of biomarkers for lysosomal storage disorders. You are encouraged to submit your questions throughout the discussion using the Q&A tab located to the right of your video player. I also want to point out that Molecular Therapy will publish a special issue on this topic, most likely in the July or August issue of Molecular Therapy, and that will soon be followed by a virtual collection that will contain articles from all of the journals of our journal family. So with that, I would like to introduce our three amazing panelists today. So our first panelist is Dr. Fabiana Perna, who is an associate member of the Department of Immunology and an attending physician in the Department of Blood and Marrow Transplant and Cellular Immunotherapy at the Moffitt Cancer Center. The Perna lab investigates how essential genetic determinants of cancer shape the cell surface proteome, thus providing targets for precision immunotherapy. She has published extensively on the translational aspects of CAR T-cell therapy, especially on the identification of novel target antigens for CAR T-cells and toxicities related to CAR T-cell therapy. She's funded by the National Cancer Institute, and her novel CAR T-cell platforms are being translated into clinical trials for patients with hematological malignancies. Our second panelist is Dr. Tristan Scott, who is an assistant professor within the Center for Gene Therapy at City of Hope, with his research primarily focused on developing protein and RNA-guided technologies, including RNA interference, gene editing via CRISPR-Cas and zinc finger technologies, towards the applications toward pathogenic viruses, cancers, and inherited diseases. He's explored a wide range of biological delivery systems, such as adeno-associated viruses and other viral vectors, lipid nanoparticles, and extracellular vesicles, which is the topic of his talk today. Dr. Scott is particularly interested in developing novel engineered EVs for delivery of therapeutic RNAi and other cargos, exploring surface-modified EVs for retargeting and using EVs to redirect cytotoxic immune cells to cancers and HIV-infected cells. And our last but not least panelist is Dr. Nicola Brunetti-Pierri. Dr. Brunetti-Pierri's preclinical and clinical research focuses on the elucidation of the pathogenic mechanisms and the development of new therapies for genetic diseases. He has a long-standing interest in liver-directed gene therapy for inherited metabolic diseases, and he has been the principal investigator of several liver-directed gene therapy clinical trials. He's professor of pediatrics at Federico II University of Naples and principal investigator at the Telethon Institute of Genetics and Medicine in Italy. I'll hand it over now to Dr. Perna, who will provide her insights on target discovery strategies for the development of future cancer immunotherapies.
Thank you, Roland, and good morning, everyone. So I'm delighted to speak on targeting tumor antigens with immunotherapy that is rapidly emerging as a promising approach for cancer treatment. My clinical and research interests are on the identification of novel targets by implementing the current target discovery strategies with the overall goal of developing novel immune-based therapeutic interventions for cancer. Molecular therapy has a strong interest in understanding the off-target effects that may result from inadvertent targeting of non-malignant cells expressing the same or similar antigens, and those represent a serious safety concern. And indeed, we are, as you said, launching a special issue on this topic. Because if all of us know that identifying therapeutically relevant surface targets is critical to ensure not only effective but also safe cancer immunotherapies, it is also true that our ability to predict the therapeutic value of candidate molecules is imperfect at this time. And while most groups, including my own, carefully assess the abundance of candidate molecules in cancer cells, it is as important to investigate the safety of candidate molecules in their evaluation in normal tissues beyond the normal counterparts before they reach the clinical trial. It is well established that the tumor mutational burden, measuring the total number of somatic mutations of the genome, influences the ability of the immune system to recognize potential neoantigens. However, hematological malignancies like acute myeloid leukemia, for instance, but also some solid tumors have a low number of mutations, and therefore the likelihood of generating neoantigens is less probable. So far, the most relevant clinical experience using adoptive cell therapies comes from targeting tumor-associated antigens, which are normal proteins overexpressed on the cell surface of cancer cells. Unlike the physiological T-cell receptor, which engages HLA peptide complexes, CARs can bind to native cell surface molecules, which do not require antigen processing or HLA expression for tumor recognition. Therefore, CAR T cells can recognize any surface molecule on any HLA background or in tumor cells that have a downregulated HLA expression or the proteasomal antigen processing mechanism which promotes tumor escape. So specifically in the settings of CAR T-cell therapy, targeting CD19 has had a transformative effect in B-cell hematological malignancies. And this depends also on the fact that CD19 has a fortunate expression profile because it is highly and homogeneously expressed in malignant B lymphoid cells. But on the normal settings is expressed only in normal B cells, causing a treatable B cell aplasia. Unfortunately, this is not the case for most tumor-associated antigens, and that is why there is a strong need, an urgent need for additional targets that can be used in CD19 negative malignancies. So the lack of suitable target antigens, in addition to genetic heterogeneity and increased recognition of the immunosuppressive factors within the tumor microenvironment, has limited the application of this promising technology to other malignancies which are CD19 negative. And I'll give you a few examples of this topic. So, for instance, in 3 patients with colorectal cancer, adoptive cell therapy with the T cells bearing a high affinity TCR against the carcinoembryonic antigen CEA resulted in cancer regression, but also severe destruction of the normal colonic mucosa because the target is also expressed on normal colon. In another case of metastatic kidney cancer, targeting carbonic anhydrase 9 resulted in unintended toxicities due to the expression of this antigen in the biliary epithelium, causing cholestasis. And finally, a last example comes from patients with breast cancer, where CAR T cells that were based on the widely used monoclonal antibody trastuzumab, after cell infusion experienced respiratory distress and displayed dramatic pulmonary infiltrate on the chest X-ray because of the expression of the target antigen on normal lung epithelial cells. So these examples serve to highlight the safety concerns regarding on-target off-tumor toxicity, which represents a significant risk in clinical trials. And therefore, we would like to highlight the need for precise protein annotation of newly discovered antigens in several normal tissues in order to inform future clinical developments. As such, as an editor of this special issue, we would like to encourage the submission of manuscripts that are focused on implementing target discovery strategies that work on standardized validation assays, and also on the engineering of novel CAR T cells, ideally multi-specific CAR T cells that have the potential of reducing the risk of on-target off-tumor toxicity by enhancing specificity for cancer cells, and at the same time may tackle tumor heterogeneity, which represents a major roadblock for all targeted therapies, including immunotherapies. And finally, combinatorial targeting also prevents antigen escape, because one of the issues in this field is the downregulation of the target antigens that contributes to disease relapse. And so this is a kind of high level picture of the field of target discoveries. My own lab is focused on developing novel pipelines that can integrate mass spectrometry and RNA-seq data from patient samples. And we recently developed also some novel computational tools for cell surface molecule annotation that we used in a project led by Francesco Di Meo in my lab that was published on Cell Reports Medicine last year, where basically we merged 5 distinct repositories for cell surface molecule annotation and created a comprehensive database that you can use to screen large lists of mass spec data or RNA-seq data in search of molecules that are highly likely localized at the plasma membrane, because we do have an interest in developing novel immunotherapies, and therefore we are looking for targets on the cancer surface proteome. So this is just an example of one of the approaches that we have utilized, and we also have another pipeline that serves to identify splice variants of cell surface proteins that may expand our space of targets for immunotherapy. And having said that, I pass it on to the next speaker. Thank you.
Hi. Morning, everyone. So I'll be giving a rundown on our topic that we're going to be covering in this special issue, which is emerging roles of tissue-derived extracellular vesicles. And so to get into the topic very briefly, extracellular vesicles, as some of you may know, is small nanosized vesicles which can be membraned or non-membraned, which are secreted from virtually every cell in the body. Because they carry surface modifications of proteins, lipids, DNA, and RNA protein, they can actually, they're being implicated in cell-to-cell communication with possible parenchyme and autocrine effects. And because they can stimulate an effect in a target cell in a target cell population, there's interest in this for biomarkers, or using them as novel ways of identifying pathways, or actually using them as therapeutics. And that's something that we're interested in. And so there's multiple types of EVs, ones that are secreted directly from the membrane. These are known as large EVs, generally called microvesicles, apoptotic bodies. And then you get small EVs, which are classically referred to as exosomes, but there's new populations coming out of non-membraned small particles less than 50 nanometers called exomeres and supermeres, and these are largely protein. But really, the EVs have been studied for a lot in cell lines and primary monocultures, and they've really been isolated from biofluids such as serum, plasma, and urine. But there's really this new, in the last sort of since 2016, 2017, this interest in doing tissue-derived extracellular vesicles. And this is directly isolating the tissues that are in the interstitial space between tissues. And so the benefits of this is really that, EVs are a packet or a product of the cell from which they're derived. And so they reflect that environment. And having a tissue where you have tissues that are having interactions with each other, they're communicating with each other, there's spatial constructs, physical cues, pH differences, all of these are going to affect the population of EVs. And this would also reflect multi-cell inputs. So there'd be a soup or a concert of extracellular vesicles, as opposed to just a single population that you would get from monocultures. And obviously there's detrimental effects to long-term monoculture that changes the population. And actually, people are finding that if you can use these EVs, you can find new pathways, new biomarkers, and also that they may even be directly usable as therapeutics and be more potent and effective than say, stem cell-derived EVs or something. And so there's really been a push to isolate these from a wide range of tissues, lung, kidney, cochlea, muscle, brain, and really with a huge amount of focus, a large, bias for cancer as well, which is what we're interested in, and adipose tissue. And this largely probably reflects just the accessibility of these tissues that you can get from human sources, such as the difficulty of getting brain tissue. And a very simple technical difference between harvesting from monocultures or from these tissues is that these are generally isolated directly from the tissues. So you either mechanically break up the tissue to squeeze out or just free up the EVs, or you chop this into a bunch of pieces and you soak it in a media that just sort of releases the EVs for a very short amount of time. You don't want to turn it into tissue culture, and just secretes into the media. And then you quickly isolate these EVs that are just being released from the tissue through an enzymatic digestion process. And then you strain these, and then you go through standard EV isolation procedures. I'm not going to go into those, but there's differential centrifugation, size exclusion, density gradient. And these might have implications for the experiment you're trying to perform. But generally, the EV isolation after that point is very similar to tissue culture. And really the important difference, I just want to highlight a couple of maybe key studies that highlights the importance of Ti-EVs and studying them in their context, is that there's a recent study that really revealed a novel prognostic profile in these Ti-EVs, which was in a colon rectal cancer cell line. So in cancer, there are macrophages associated with cancers, generally referred to as tumor-associated macrophages. And this was a study in a colon rectal cancer cell line. And tumor-associated macrophages are generally considered a negative marker of progression. They're immune suppressive and pro-tumor. But these authors did a really great study where they actually, in order to identify specifically the population of EVs coming from these tumor-associated macrophages, they used an antibody to the tumor-associated macrophage and eliminated them in one set of the tumors. And so through sort of a negative analysis, they could reconstruct the profile coming from the tumor-associated macrophage when they isolated this soup or this concert of EVs coming out of the tumor. And what they found was really interesting. When they did proteomic analysis on the macrophage itself inside the tumor, they found, as expected, this is an M2 or tumor suppressive macrophage. But when they actually profiled the EVs, they actually found that these were M1 or pro-inflammatory EVs. And they demonstrated that these could actually stimulate T cells, they could change the lipid profile of the cancer, and essentially create a situation where these were actually being a positive prognostic marker. When they compared the profiles of the cells versus the EVs, they found that to predicted outcome databases, they found that the EV profile was actually more significant and correlated with the patient's survival within the tumor. So you have got this disconnect between this remarkable disconnect between the cell and EV profile, demonstrating that if you can expand this, you might be able to actually get more prognostic information in patients. And then there's also the case of trying to find new markers of communication between different tissues. And it's well known that obesity, and so in a separate example, it's well known in obesity that this results in chronic inflammation and a higher risk of cancer. And there's generally a cancer switch from mitochondrial respiration to aerobic glycolysis, which is called the Warburg effect. But people are actually finding that in some aggressive cancers, this may actually revert back to mitochondrial respiration. And this might offer new opportunities of targets if you can figure out how this is happening. And in a study, they actually is looking at breast cancer-associated adipose tissue, so fat tissue, from obese as well as lean women. They were actually able to show that the Ti-EVs isolated from the obese fat tissue on a breast cancer cell line was actually increased proliferation. And through proteomic and RNA-seq analysis, they actually found that there was transfer of small regulatory RNAs called microRNAs, which was actually converting the profile to this more respiratory and mitochondrial-dependent process. And actually, by blocking these microRNAs coming from this adipose tissue, you could actually get the proliferation. So this offers a new means of blocking a profile coming from a different tissue source, which is enhancing your cancer source, offering new therapeutic targets. But really, in terms of switching gears a little bit, in terms of biomarker discovery, this is having a huge impact on trying to validate liquid biopsies. And so you've got, there's a huge interest in using extracellular vesicles from serum to isolate them in order to diagnose cancers, because the cancers should secrete these into the bloodstream, and we should be able to isolate these and find the profile of the cancer cell. The problem is in the serum, you've got basically every organ contributing to the EV profile, and that signal gets vastly diminished. And so the rationale is using Ti-EVs, or these tissue-derived EVs, in order to find factors that could validate bona fide serum targets. And through a great study, through a huge study through the Leiden group, they did proteomic analysis on many, many different explant tissues, as well as cell lines and etc. And they of Ti-EVs, and specifically from lung and pancreatic tissue, and they isolated tumor tissue as local healthy tissue, and in the case of lung cancer, distant tissue. And then they found, basically, they reconstructed the markers that were not only enriched, but also exclusive to each particular cancer. And then they correlated this with serum from cancer and healthy patients. And they actually be able to come up with potentially a predictive panel that could be not only determine the specific cancer type, but in some cancers, in a percentage case of cancers, you don't actually know what the original tissue is. And you can actually then find out that a cancer of unknown source, which would have massive prognostic as well as diagnostic implications for those patients. And so just to conclusions, the Ti-EVs are emerging as this really new paradigm in EV research. It's really offering unique insights that cannot be observed in monocultures or cell lines or even in serum. They can be used to validate liquid biopsies. Something that I haven't gone into is identify novel biomarkers, which would not be present in monocultures due to the lack of that environment, the tissue environment. And also finding novel pathways for prognostic as well as therapeutic intervention. And actually, people are now also looking to use Ti-EVs, actually isolate them and apply them as a therapeutic intervention, which is one of the main focuses of my research group. Thank you.
And I think I will go next with a different topic because I will be talking about biomarkers for inherited metabolic diseases, and particularly for a subgroup of the inherited metabolic diseases that are the lysosomal storage disorder. So this is a group of diseases that is typically treated by either enzyme replacement therapy or allogeneic hematopoietic stem cell transplantation. But both approaches have limitations, and they both leave substantial disease burden, particularly for brain and some systemic manifestations. And for many years, lysosomal storage disorders have been considered good targets of gene therapy because the ineffective treatments that is currently available, and also because of the specific biology of the lysosomal enzymes that can be secreted by a factory organ and uptaken by the mannose-6-phosphate receptors by distant organs, allowing the cross correction. And in the last decades, there has been really a huge increase in the number of clinical trials for gene therapy, and of course, the evaluation of the the efficacy of the experimental treatment is critical, and this is typically done on clinical endpoints. There are several well-established endpoints that, of course, can be used, including survival, cognitive and neuromotor function, endurance, growth, joint mobility. However, most of the evaluation of these endpoints is challenging. And the main limitations of these clinical endpoints is that they would require long follow-up periods in a large number of patients. So this, of course, is challenging, especially in the context of genetic diseases in which enrollment of patients may be an issue. And availability of biomarkers as a surrogate endpoints rather than the clinical endpoints, of course, has a great potential particularly for accelerating the drug approval. And indeed, there have been already examples of approved drugs that in which the efficacy has been based on surrogate endpoints, like in Alzheimer's disease, for example, the reduction in the amyloid load evaluated by MRI, by brain MRI, has been the basis for the approval. And similarly, we can think about also some type of biomarkers for lysosomal storage disorders that can accelerate approval, and particularly when the possibility of having long-term data is challenging. So the ideal biomarker needs to have important features, such as has to reflect the disease burden in all the body districts that are affected by the disease. The concentrations of the biomarkers that we measure should be well separated from the concentration of healthy controls. And also, these concentrations should be rapidly changing in response to a specific and effective treatment. Of course, and one would also like that the biomarker can be measured reliably, rapidly, and inexpensively in body fluids that are collected by non-invasive procedures. Of course, having all these features is impossible. I mean, there are no available biomarkers that can meet all these requirements, and the currently available biomarkers have some type of limitations in one way or in another. And in the biomarkers for lysosomal storage disorders are based on bioimaging studies, like brain MRI or the liver and spleen sites, for example, but also markers that can be measured in body fluids. Like one of the most commonly used biomarker is the glycosaminoglycans for lysosomal storage disorders. So these are macromolecules that are made by long, linear, and negatively charged chains of repeated disaccharides. They are components of the extracellular matrix, and the increase in the glycosaminoglycans is the hallmark of several mucopolysaccharidosis that is a subgroup of the lysosomal storage disorder. And basically, the glycosaminoglycans are the substrates of many of these enzymes, and therefore the enzyme deficiency results in accumulation of the substrate, and if there is any improvement of the any gene transfer that can provide the functional enzyme, there would be more degradation of the enzymes, and one would expect to see a reduction in the concentrations of the glycosaminoglycans. There have been studies on natural history of some of these mucopolysaccharidosis that have correlated the concentrations of the glycosaminoglycans with the disease progression, the morbidity, and long-term survival. Overall, the glycosaminoglycans has features of a good biomarker. But what we really measure is mostly urinary biomarker, the urinary GAGs, that has been historically one of the most commonly measured biomarker for mucopolysaccharidosis. And what we measure is typically the glycosaminoglycans that has been accumulating in the renal cells that are then falling off from the kidney into the urine, and so we really measure by urinary glycosaminoglycans. We measure the accumulation of these substrates in the kidney parenchyma. But nevertheless, there have been studies showing that the urinary glycosaminoglycans correlate well with the degree of accumulation of GAGs in other tissues, and this has been, of course, done in animal models. But more importantly, there has been a good correlation between the reduction in the urinary GAGs and the response with several enzyme replacement therapy. So with the a response with the currently available treatment for the lysosomal, for several lysosomal storage disorder, that is the enzyme replacement therapy. Based on this historical data, the urinary GAGs continue to be used for monitoring the efficacy also on gene therapy. But of course, there are there is the concern that urinary GAGs may not fully reflect the accumulation of GAG into tissues. And also, we can probably get better with the determination of GAGs, because the urinary GAGs is a mixture of different glycosaminoglycans. And it is possible that if we measure the more specific types of glycosaminoglycans, we can get more precise in the determination of the efficacy. And in fact, there are methods that have been developed with using tandem mass spectrometry to detect the specific glycosaminoglycans in the urine, as well as on blood, or even dried blood spots. That it's even more convenient way to measure a biomarker, because it makes things much easier in terms of simplicity of the collection and transport of the samples to the laboratory coming from multiple sites. For example, multiple sites participating into a clinical trial. However, one of the limitations is that while we do have data on enzyme replacement therapy and the urinary GAG, showing that a good correlation, the measure of the specific GAGs by tandem mass spectrometry has not been really validated, for example, there is not enough data in either in patients with enzyme replacement therapy or other treatments evaluating how good are the specific GAGs in reflecting the disease status of several mucopolysaccharidosis. So we do have GAGs that are a relatively good marker, have been used for enzyme replacement therapy, and they are currently used for several gene therapy trials. But of course, there is the need of other additional biomarkers. And one of the advantages of the lysosomal storage disorders is that we can actually measure the actual enzyme that is the product of the gene transfer. And this actually, in contrast to several other diseases, for example, spinal muscular atrophy, for example, we do not measure the efficacy of the gene transfer. But in the case of lysosomal enzymes, because these are secreted naturally by the producing cells, we can actually measure the lysosomal enzymes in the blood, and this is a direct readout of gene transfer efficacy. But besides the traditional markers like GAG, urinary GAGs, glycosaminoglycans, and lysosomal enzyme activities, there is lots of research going on more advanced or more sophisticated biomarkers that can reflect rather than the primary storage, the consequence of the primary defects. For example, there are several technologies that have been that are under development as biomarkers based on mass spectrometry, metabolomics, lipidomics, even proteomics, transcriptomics and also to look at inflammation, signaling pathways. All these are technologies based, again, on the measurements of the secondary consequences of the storage, because the storage causes lots of changes in the cell that can eventually be analyzed by all these high throughput technologies. Of course, this also makes the challenge of interpreting a multi-dimensional data sets and extract from this huge amount of data, the data that will be correlated with the clinical status. That is an open challenge in the field, of course. With that, I think I run out of time, and we can move on to the next, to discussion, I guess.
So I'm hoping that the audio is given back to me. Is that working? So so first of all, thank you all three of you for these informative presentations and food for thought on all of those topics. I can almost see Tristan kind of being the anchor of this, of a grand collaboration between all three of you, because I'd imagine that these extracellular vesicles will be useful to develop biomarkers for storage disorders and obviously in diagnosis of cancer, as you already pointed out. So, and we have a couple of questions here about those topics in the chat. One is, I guess, directed to Tristan, which is what stages of cancer can be effectively diagnosed using extracellular vesicle-based methods, and how do these methods compare in early detection versus later stages of cancer? Maybe you can comment on that.
Yeah, no, I think it's a great question. I think it's early days for using these as liquid biopsies, but as I mentioned, if you can find a bona fide EV marker that you can pull out and enrich for, you theoretically could diagnose it at very early stages, even when each tumor is not even visible. And there's examples of this where they've had resection surgeries where tissue's been removed, and when isolating, EVs from what is supposed to be healthy tissue, they still get the same tumor and inflammatory profile, and this correlates with a return of the cancer. So even when you have no detectable cancer present, you can still detect the cancer EV profile. And so this should theoretically be able to detect at very early stages if you can really pin it down. And generally the marker correlates with the stage of disease. So you can actually get the correlation of how much in the serum you're detecting and how much you can detect in the Ti-EVs. So stage 4 versus stage 1 will have a linear correlation between the markers. So it should, if you can pin them down, it should be a reliable and sensitive marker.
Yeah, that's actually quite impressive. So you talked about how these EVs contain sort of signatures of a stage of the disease, a lot of it related to the immune response to the tumor. Do these EVs potentially contain tumor antigens that can be identified that could be useful for Fabiana, for example?
I'm certain there would be. Yeah, definitely it picks up many components of the tumor and generally reflects the tumor state. And so I think you could use it, definitely as a marker for the tumor, but also potentially as a marker of responsiveness. I don't want to overextend it, but I think the profile would change if you're actually getting a positive effect. I'm trying to think of an example off the top of my head for what I've read, but I believe you should be able to do that. Yeah, but definitely it'll be enriched for these tumor antigens, and that's part of the mechanistic transfer. And often it's happening between the immune cells and between the cancer. It's not a one-dimensional one transfer, but you can often these EVs are now being implicated in augmenting the immune states and also blocking immune responses. So antibodies, and if you can eliminate these EVs or design smarter drugs to target the tumor cells, you can actually get around a lot of those tumor-enriched EVs. Theory.
So I have a question for Fabiana, and then there are actually a host of questions that have just come in for Nicola. So we'll get to those in a second.
I was going to ask about the gag.
Fabiana, I was wondering, you talked about the need to identify tumor antigens, novel targets, and how you have diversity even for the same type of tumor from one patient to another, and how things can change over time in the same patient. And so you concluded that you need to identify these potential new targets and have a sort of a diverse approach to maybe targeting more than one, also, to deal with tumor specificity. It all sounds very complex. So, but I could envision that maybe one could have sort of a toolkit eventually available that based on your findings of what these various antigens are, and then one could perhaps more rapidly decide on which combination of CAR Ts or antibodies and so forth one can use. But that it's not a traditional way of how the regulatory agencies, right, traditionally view drug development, where you have one defined drug, and then if you switch to another, you would have to do a whole clinical trial. So obviously that concept wouldn't work. What's your experience with this? Because that really changes potentially what you're doing will change the speed also with which you can intervene in a patient and make it more of a precision medicine. But that would require flexibility, right, by the regulatory agencies.
Yeah, your question is very important and right on topic. I mean, going after precision therapy, right now, I'll give you the example of AML, where over the past year, there has been a great understanding of the molecular mechanisms driving this disease. So we know that right now, there is a complex clonal heterogeneity where multiple clones are genetically defined. So it is unlikely that one target will be able actually to target all clones of one disease, and even there are different subsets of leukemias, so it's just not one AML. So I think ideally this is what the field is going after, is basically having a kind of a toolkit of a potential multiple combination of platforms that can be provided depending on the expression of a specific target and also specific genetic background. And a little bit, I mean, even if the therapeutic options for AML, just to remain in this example, remain limited, because over 40 years we still use a chemotherapy first line. So, however, because now we understand more the different mutations driving this disease, it is likely to envision a future where also in the immunotherapeutic space, based based on the specific genetic driver, we can actually propose one combination of targets compared to another. And they don't need to be necessarily cell surface proteins. It could be, I don't know, a CAR against a surface proteins combined, for instance, with a TCR against a specific peptide. And there are preclinical examples that are coming out recently. Now, to address the issue of the regulatory process, that remains very complex. So the FDA puts lots of efforts on the safety of each novel drug, and so it is important. I think, in principle, we can always show efficacy of, at least in the preclinical stage, of these therapeutic options, but it is important to address the concerns of safety that are not exclusively derived from on-target off-tumor effects, as we just discussed, but they can derive from the genetics of the tumor, from the CAR constructs. So independently on the target, different CARs can lead to different toxicities, the contribution of the tumor microenvironment, and so on. And so it is quite a, there are multiple layers of complexity, but I think if we can go one step after another and perhaps selecting suitable targets which are biologically and therapeutically relevant, and then adjust the CAR platform, just to give you this specific example, based on the type of antigens, and then matching potentially the genetics of patients, we can move forward this beautiful concept that I think all as a scientific community would like to embrace for patients.
All right, I think that makes perfect sense. So now it's, I think, Nicola's turn. So let's see. Are there any notable differences in the biomarker profiles for storage disorders in neonates versus pediatric patients versus adults? How does that change with age?
Yeah, you would. So in patients that are untreated, you would expect major changes in the biomarker, simply because patients that are with adult patients are expected to have milder form of the disease because they, untreated, they survived into adulthood, whereas children, the earlier the presentation, the more severe is the phenotype, and therefore the higher is the concentration of the biomarker in the body fluids. This is what you would in general expect. Of course, with the treatment, everything changes, and depending on the response to treatment, the profiles may be affected as a consequence of the treatment, of course. And the next question is about newborn screening. I read, and of course, yes, there is newborn screening. Newborn screening for lysosomal storage disorder is possible, and indeed it has been implemented in some countries. So the technology for diagnosing newborn with lysosomal storage disorder is definitively available.
Yeah, I think, and then there were there are a couple of questions that have to do with the how accurately some of the methodology is that you described, and also how specific the interpretation of those biomarkers is, as opposed to, picking up a signature that may actually reflect different other diseases. How specific are those methodologies and the interpretation of the results?
Yeah, that's a great question. However, so when we use the biomarker, and typically we use it for monitoring treatment efficacy rather than diagnostics. Of course, it is very possible that some of the molecules that we measure may have may be falsely increased if there are other ongoing conditions. Of course, that's an issue at the diagnostic level, when we are using it for diagnosis, or in the case the patients with the lysosomal storage disorder, in which we are measuring a biomarker, has a comorbidity of another disorder. But it is very well possible that some of the biomarkers, particularly those that are acting downstream the storage effect, so those that are measured as a consequence of the storage, may be somehow promiscuous. So there may be different pathological processes that can increase in a non-specific way, for example, cytokines or specific metabolites, some specific may result in specific lipidomic signature, for example. In contrast, the GAGs, the old metabolite, it's quite specific. I've not heard about specific non-genetic conditions that are associated with a false increase in urinary glycosaminoglycans, for example. There could be, but I think that would be rare.
There's a related question, which I think you've already started answering, which is about the disease, how accurately these biomarkers can be used to reflect disease burden. And you already mentioned how, somebody with milder disease will obviously have a greater chance to progress to adulthood versus a child who is born with severe disease, and so how accurate are biomarkers in reflecting disease burden in a patient?
As I mentioned, they are not perfect. The biomarkers we have, they are not totally reflective of the disease status, but somehow they are still acceptable, because if you look at for example, enzyme replacement therapy, that has been approved because of a reduction on the glycosaminoglycans that correlated with clinical improvements. Whether the improvements was a consequence of the reduction in the glycosaminoglycans, I think is still to be demonstrated. But overall, natural history study and the enzyme replacement therapy study would suggest a good correlation between the clinical status and the concentration of the urinary GAGs in urine, for example.
I think that the last question I see here on the bottom question I see on the Q&A is that's obviously a hot area of discussion, right? For each individual disease is biomarkers as primary endpoints as opposed to clinical outcomes. So one versus the other. Can you comment on that? What's the experience been so far with this? I'm sure there are some sticky points there. So one of the challenges, particularly with like lysosomal storage disorder, and as well as other genetic diseases, is that if you want to look at clinical endpoints, it may take several years to get an answer about the efficacy of the trial, and that makes it difficult to run clinical trials. On the other hand, I think we need better biomarkers that we know are correlating better with the disease status to get approval based on biomarkers. But anyway, this is coming because now there have been examples in several examples now of drugs that have been approved based on a biomarker that we know are correlating very well with the clinical status. That has been the case in some of the liver fibrosis, for example, that field in Alzheimer that I mentioned, in chronic
kidney failure. Not people have looked at GFR as a biomarker that we know correlates very well with the clinical status, and on that basis, drug have been approved. So I think it's a discussion that depends on the biomarker and on the disease and the knowledge available for that particular biomarker and disease.
There is also another question for Tristan about standardization of diagnosis of cancer progression using EVs. Is that how far has that gotten so far? Do you have a sense of that?
Yeah, I think it's still early days. I think it's set for the same standards, I think, as any diagnostic platform: sensitivity and specificity. I think what I didn't, just because the brevity of this talk, which we go into more in the special issues, is the challenges of EVs as this, and a lot has to do with purity of the system. So there's many ways to purify EVs, density gradient, ultracentrifuge with increasing levels of complexity. But a lot of the current studies have been done with just standard differential ultracentrifugation, which actually has a lot of contaminants, non-vesicular contaminants in them, which are not actually EV based. And these are undoubtedly biomarkers, but whether they're EV biomarkers would need to go through further validation. And if you can just find a lot of studies have found surface markers of EVs would be, because then you can pull out the signal and more readily pull out the signal to detect it. And there's ones like UZU1 and a lot of mitochondrial proteins, because cancers tend to upregulate their mitochondrial proteins, and there's actually implications for that for disease progression as well. And these are surface markers, and you can easily just sort of pull these out. But these are also suffering from sort of specificity issues, where you'll get as I see it now, based on the literature, you'll get a lot of false positives. It's upregulated, definitely in cancer scenarios, but you would still detect these in sometimes benign cases, such as ovarian cysts versus ovarian cancer, which is not, you want to be able to distinguish between those two. And some people have actually done some really great studies where they've actually tried to find markers that have lineages. So they take they don't just do healthy versus tumor. They take the healthy tissue and then they use the proteome of the healthy tissue and they compare it to the cancer tissue, and they try and find common markers, but one that shows progression from the healthy tissue to the cancerous tissue. And you can then actually strongly delineate between that and any other benign scenario. But yeah, these are some of the challenges that the field has to face. But I think the standards would be similar to any diagnostic platform.
I would also like to point out that the special issue will contain a review article from your collaborator, Dr. Kevin Morris, which will comment on using EVs for therapeutic delivery of RNA molecules such as RNAi or non-coding RNA, which is also a novel concept that is just emerging, but a hot field of research. So we've come up towards the end of the hour, so I want to thank the attending. I would like to thank everyone who attended today's webinar, and of course thank our panelists for sharing your expertise and for the great discussion. I'd like to thank everybody who posed questions that have been excellent grounds for discussion and dissecting of these issues. I would also want to mention that this event was recorded, and yet will receive an email with a link to watch the recording when it becomes available. And please continue to explore cutting-edge research, emerging therapeutics, and cell and gene-based cancer treatments at ASGCT's Advancing Gene and Cell Therapies for Cancer Hybrid Conference, which will take place on October 16th and 17th. Learn more about this at asgct.org, or by visiting the upcoming events tab. Finally, ASGCT values your feedback and welcomes suggestions for our future sessions. Please take a moment to complete the survey in the tab to the right of your video player, and thank everybody again. Thank you so much.